Okay. Dear audience, thank you very much. Thank you, Max, for inviting me. This is a great honor to be here on the conference of Gerophysics. It is actually a conference of my dream. Actually, when I started to do computational biology of aging, I really dreamed about this conference, and I'm here. I'm on short talk. I want to I want to discuss with you quite interesting and important topic for the modern machine learning, which is called uncertainty. My title is Estimating Uncertainty in Biological Age Prediction, and I should prove you today why it is a fundamental challenge behind biological age. Okay, we start from a kind of average definition of biological age, which can be averaged across different definitions of biological age exist, and we can treat it as a number that signifies a person's health. Of course, it's related with chronological age and age acceleration term through this simple relation. Typically, three motivations exist behind biological age. People want to have a measure, a generalized measure of a person's health. On the other hand, it may serve as an endpoint in clinical trials of geroprotectors. And in fact, if we have a good formalism of biological age, we could even use it for finding new geroprotectors in silico. On the other hand, we predict biological age from biomarkers. This is denoted here as X with some function f. And by simple definition, this function f is called aging clock, a function that predicts biological age from biomarkers. And different aging clocks exist. If you use DNA methylation for X, this is the f will be epigenetic clocks, and corresponding B is epigenetic age. And for the same for transcriptomic clocks, if you use gene expression as X, and so on. For blood biomarkers, of course, it's blood clocks, and B is called biological age. Very simple. However, we have a problem. Biological age cannot be measured directly. It's not a physical entity that you can just measure from somewhere. So different scientists propose different approaches to do that, to formalize a kind of measurement, but actually the predictions of biological age. First generation clocks, when they appeared, they utilized the idea that prediction error in first generation clocks. Prediction error in first generation clocks is in chronological age regression. It can be treated as an age acceleration term. There exists another type of clocks which are unsupervised. They use the idea that biological age can be interpreted as a latent variable driving biomarker dynamics. And of course, second generation clocks, which use the idea that biological age is some convenient representation of all-cause mortality hazards. In all three cases, you see the prediction error is here. The prediction error in first, second, any of this model actually has prediction error because it's the core of statistical inference that you always have a noise in your data, and your model cannot be perfect. It faces with some, it contains error term. And a simple example that demonstrates why this error is important is the following example. Suppose you have data and you trained your favorite aging clock model. You fit your model to the data, nice fit obtained, and you received a new patient which stays far from the training data set. And you predicted the age and obtained the age 38 years. Okay, if you're suspicious enough regarding this prediction, you might ask, okay, what is the error of my prediction in terms of standard deviation? What is the uncertainty of my prediction? And to answer this question, we need to first understand the concept of so-called out-of-domain prediction and understand the taxonomy of uncertainty. Let me start from the simple example. What is out-of-domain prediction, suppose you have a simple model that you trained to classify images of cats and dogs. It can be just neural networks that classifies this. You trained this model, received nice metrics, but during testing time, you obtained something new. You obtained an out-of-domain example. And if we try to classify this with the same model, of course, we understand, because our general intuition is great, we understand that this is an incorrect problem statement. On the other hand, coming back to the realm of epigenetic clocks, we can see the similar situation. We train our regression model on the epigenetic profiles of healthy people, and then use the same model for prediction something absolutely maybe different, like dynamization of reprogrammed cells. Will this prediction be out of the main one? This is a good question, and now we are ready to understand the taxonomy of uncertainty, because if we try to predict the out-of-domain patient, coming back to this example, we actually may split the uncertainty into 3 parts. The first part is a so-called aleatoric uncertainty, related to the internal data noise, epistemic uncertainty, which relates to the lack of knowledge about the actual new patient which you want to predict. This is an epistemic uncertainty. And model uncertainty, which depends on the class of models you used for fitting your data. These 3 types of uncertainty is a typical classification from machine learning articles. And when you try to predict this new patient, which is out of domain, out of distribution of this, what you actually obtain is a kind of generalization of hallucinations, which we probably can face when used LLMs. How to tackle the uncertainty? Biological age cannot be measured explicitly, as I said. Okay, we cannot explicitly test the correctness of predicted biological age, but we can use two approaches to test the uncertainty, to control the uncertainty of our prediction. First, we can check the dataset shift. We can check if the test dataset is not related to the training dataset. Is there a shift in covariates between 2 datasets? For example, these samples are out of domain regarding this one. And the second, we can use some aging clock models, machine learning models, which can estimate their own prediction uncertainty. And let me demonstrate the most striking example of out-of-domain prediction, which I elaborated more during last time. This is an example of reprogramming-induced rejuvenation prediction. It was suggested that reprogramming rejuvenates aged cells. It was also suggested that this rejuvenation can be measured by epigenetic clocks, specifically first-generation epigenetic clocks or Horvath clocks, for example. And this looks like this. Epigenetic age decreases during the process of reprogramming. This result was repeated and reproduced a lot of times. And moreover, even me, and with my collaborators, specifically with Vadim Gladyshev group, we reproduced this result by training transcriptomic aging clocks and applying this to the transcriptomic reprogramming data, and also obtained this decreasing pattern and good correlation between epigenetic and transcriptomic clocks. But okay, why two different clocks and different modalities receive the same result of decreasing in age? I thought a lot of time about this, and my best explanation is the following. What actually clocks catch when they predict decreasing pattern of epigenetic age is a kind of projection of predicted age on the axis of aging. In fact, reprogramming moves cells to the some third unrelated and out-of-domain state. But the projection of this reprogramming to the axis of aging looks like rejuvenation. And this is what actually aging clocks catch. And to confirm this, we tried to use the previous two rules I mentioned for tackling for controlling the uncertainty. And what we checked first, we looked at the data set shift between aging data set and reprogramming data set. We used this aging data set to train aging clocks. And then, yes, we see that the reprogramming data set is out of domain regarding the aging clocks. And the second thing, we use uncertainty enriched models to predict the actual confidence intervals of the predictions, and they were super large. So, take-home messages and conclusion. First, we need to be aware about the uncertainty regardless we model biological age, system resilience, or individual biomarkers, where we use machine learning models for prediction. 3 simple rules for tackling the uncertainty. First, check dataset shift, if you can do this. Second, use model-enriched, uncertainty-enriched machine learning models for predictions. And third, and maybe even the most important, think twice if what you want to predict is a coach relative to what you trained on. For example, cats and dogs. Thank you very much. My colleagues helping. My gratitude to my colleagues who helped me to achieve this state for now. And I will be happy to answer questions if I'm not super out of time.
Thank you very much. So we have time for questions. Maybe I can start. So you suggest that the idea of the cells, when they reprogrammed, let's say, from somatic cells to induced pluripotent stem cells, might not be quantified with epigenetic clocks. Why is that then when we observe, when we quantify biological age of cells for many species, like human going to IPS, or mouse going to IPS, or pig going to IPS, cells reprogrammed, all kinds of different cells, they consistently show the age approximately 0 based on the clocks. And we know that the cells are literally young because we can generate many cell types from them. We could generate whole animals from them. So they seem to be young, and the clocks seem to show that they're young. But then you say that the clocks cannot be used for that purpose.
Yeah. I want to emphasize that I'm not claiming that there is no rejuvenation during reprogramming. Possibly it is there, and moreover, if we analyze transcriptomic signatures, for example, or if we analyze, by the way, yesterday we see that reprogramming actually can slightly increase the lifespan. Okay, there is something, there is a rejuvenation, small rejuvenation signal. The only claim I do is that the clocks is an unreliable instrument for testing this rejuvenation. My claim is we need something else. We need another instrument for measuring this rejuvenation. Maybe this should be much more complicated instrument which tests a lot of different biomarkers to catch actual rejuvenation. But, and this is the only claim. Clocks are not okay here because of the epistemic uncertainty.
Yeah, thank you. Brian?
Maybe naive, but you mentioned it as a projection, but that projection seems to capture some of the directionality of the rejuvenation. So is there an avenue here to look for the commonality of changes and the discontinuity of changes that go with reprogramming versus, measuring the clocks to try to understand what's going on? The reason I think we all talk about this idea of, oh yeah, it's possible to live forever because the species lives forever. We can make a new rejuvenated cell that becomes an individual. And some people think of that's evidence that aging might be reversible. Other people are like, well, it's a different phenomenon. But maybe there's a way to look at this more mechanistically and dig in and try to figure out what things are in common and what things are different. Does that make sense? Sounds like a naive question.
Sir, could you shortly repeat your question?
Or rephrase.
I'm sorry.
Are there elements of rejuvenation that you can get from the clock when you reprogram cells and then elements of discontinuity. In other words, it looks like the projection has some similarity, but is not a return of the clock. And so if that's true, can you dissect those at the methylation level or whatever clock you're measuring to try to figure out what mechanistic components are actually are part of rejuvenation and what part is part of aging that's not cellular rejuvenation?
Okay, let me give another striking example why possibly the projection itself is not enough to take a reliable prediction. Suppose, and I'm sure I saw the article where people tried to use epigenetic clocks to the cancer cells. And they found that cancer cells are younger than just the same cells of the same age. And this shows us that the prediction of cancer cells with epigenetic clocks is also an out-of-domain prediction. And we understand that this non-desirable physiological state.
Yeah, but is it a younger physiologic state? That's a different question. A cancer cell.
Yeah, yes, but I'm saying that prediction of cancer cells is an out-of-domain prediction also. And we may, if we will consider cancer cells with lower age as the actual rejuvenation, we are kind of in a trap, because cancer cells can be seen as a rejuvenated one in terms of clock's prediction. But we should understand that this is unrelated third state, which is not physiological and undesirable for us. And possibly the reprogrammed cells is also undesired state, physiologically undesired states, which is dangerous for us. And we should avoid interpret the rejuvenation in out-of-domain samples because it is unreliable predictions.
Thanks.
Well, I thank you for bringing up this important issue to the field. I actually fully agree that uncertainty is an important challenge. Thank you very much, and thank you everybody for attending.
Thanks a lot.